And ladies and gentlemen, our next lecture will be delivered jointly by Professor Jaroslav Štěrba and then by Dr. Regina Demlová from Masaryk University and University Hospital Brno. First will speak Professor Štěrba, a professor of oncology and pediatrics and head of the pediatric oncology department at the University Hospital Brno. Then Dr. Demlová, she is associate professor of clinical pharmacology, director of CREATIC and chair of the ECRIN Network Committee. Their presentation is titled Biomarker-Driven Adaptive Treatment Strategies for High-Risk Pediatric Tumors. In this lecture, they will address one of the most urgent challenges in pediatric oncology, the poor outcomes of children and young patients with high-risk or relapsed cancers. Dear Professor Štěrba, we are looking forward to your presentation.
Still good morning, ladies and gentlemen, and let me take you to the small part of oncology, just about 1%, but much more complicated than we could hear from Professor Rojas. Those are kids. Those are my mandatory disclosures, as I am wearing two hats, similarly like Professor Rojas. I am a clinical pediatric oncologist and at the same time member of pediatric committee of EMEA, making my split realistic. So what's the current situation for children with cancer in Czech Republic? You can see that over time our results are improving, and there is no difference between survival of Czech kids and kids in France, Germany, Austria, or Spain. And it's reflected recently that in Europe there is over 800 pediatric oncology centers. 100 of them, they are labeled as a European reference network, and just 68 of them, they are selected as something like a Champions League for soccer as ITCC centers, and both Czech centers, Prague and Brno, are member of this elite club. So what's the problem? What do we want? We should remember that it could always be much worse. So what is the current pediatric oncology legacy of issues? Our survival has plateaued during the last 10 years. We are still losing children, as you can see from this from those curves recently published and assembled by the Benchista European initiative. Still, we are losing children with high-risk pediatric tumors. And as you can see, unacceptable side effects from this is 20 years after treatment for low-grade glioma, and the quality of life of with such spine won't be very high. So unacceptable side effects, late effects, secondary cancers, and contrary to adults, we have to wait for our CAR T cell approval or approved CAR T cells after failure after allogeneic bone marrow transplant. On the other hand, our patients and parents, they want safe and effective treatment now. We have better understanding of tumor biology, and it seems clear that histopathology is just giving us probable diagnosis. But the access to whole genome, whole exome sequencing for everyone, RNA-seq methylation, which is a must for children, it's not available everywhere and for everyone. Historically, kids, they were considered as a vulnerable group that should be protected from research. Recently, we realized that clinical trials are necessary to ensure that we know what we are prescribing and having good level of evidence. And children are best protected through research, not by shielding them from research. However, the design of good clinical trials in pediatric oncology is more and more challenging. For medulloblastoma, we have 18 plus subtypes. Just for a rare ATRTs, we have more than 5 subtypes. So The reality is that who is actually protected from pediatric research? Current clinical trial design, which was originally introduced to protect public from unfounded claims or severe adverse events, over the time evolved in a rigid structure, which is unsuitable for new biological agents like we heard about the CAR T cells. And standard is to give one single new agent to standard MTD, which is something we are not happy with. If we just realize the number of newly approved oncology indications for adults and kids, we see that the numbers are absolutely uncomparable. During the whole history of oncology, there is just 100 approved drug for kids, and for adult world, there is over 300 approved from 2022 only. So the reality, what is the reality on the ground? Let me take you with one of my patients with CAR T cell story. Abdominal Burkitt lymphoma being treated by standard protocol chemoimmunotherapy with anti-CD20 antibodies, early good response, however, progression on treatment. CAR T cell participation in a trial was requested, but the kid didn't have measurable disease, just evaluable disease. So we were put on hold until the measurable disease evolved. We got a permission to enroll the kid on a CAR T cells trial. The CAR T cells were infused. However, rapid retroperitoneal progression was seen 2 weeks, and later on, we gave a second CAR T infusion with another progression. So what does it mean? Are CAR T cells ineffective? Despite having CAR T cells in a circulation and available in a bone marrow, you can see the clear signs of progression. And assessing whole exome sequencing, there is almost no change between initial diagnosis and further progression. What does it mean? The gene-centered cancer paradigm, which is prevailed almost 100 years, isn't valid here, doesn't work. What we have seen in a repeated biopsies, no difference in exome on a DNA level, just minor shifts on a transcriptomic level, but we have seen marked evolution of the tumor microenvironment, raising number of double positive CD163, CD68 cells, which means M2 macrophages number was rising in the tumor lamps despite having in a circulation still detectable CAR T cells. So the kid started metronomic chemotherapy plus thalidomide, and within 10 days, we could observe fantastic regression of all tumor lesions. So what could be the interpretation and the lessons learned? Myeloid-mediated immune suppression is protecting Burkitt lymphoma cells from effectivity of CAR T cells. Linear gene-based explanation of cancer development is not working here. Can macrophage M2 reprogramming is possible? There were over 200 possible reprogramming agents investigated in more than 700 clinical trials without any market success. So how it comes? And recent clinical trials, we are dealing with highly dynamic diseases, and they are treated with just fixed, non-adaptive protocol. They are driven by pharma with filing intention and by single agent approach. From the company point of view, the company producing CAR T cells, does it mean CAR T cells were ineffective? From the company point of view, they failed as a single agent, but most probably they contributed to the overall response if used in a combination with just simple metronomic treatment. My understanding is that drug-centric approach is highly detrimental for potential combinations with other immune therapies or flash radiotherapy, which is making huge sense recently, being combined with immune therapies. And there is an importance of biomarkers, or rather set of different biomarkers, preferably from peripheral blood, because we cannot biopsy everyone. So not just the cancer cell, we have to understand and think about the tumor microenvironment as a highly dynamic ecosystem, and genomic and tumor microenvironment information should guide our therapy approach. Targeted immune therapies, they are designed for adult diseases. And in a pediatric, our pediatric applications, they come from adult world. There are no specific pediatric studies, despite we need to know safety and efficacy. And different etiology and biology, so there is a different degree of extrapolation. There are some good news because we understand more the biology. There are some preclinical platforms, but they are not game changers. There is an individual tumor sequencing routine in Europe, but it's a solution just for low-hanging fruits, roughly 7–10% of our patients. So what are the major issues in pediatric oncology recently? It is already mentioned drug-centric single-agent approach with filing intention and missing combinations, dynamic tumor ecosystem, which are targeted by fixed non-additive approaches, and it's too easy for pharma to get waivers. Just recently, during coming days, PRAME glypican pediatric development was proposed inside the EMA, requested by the PDCO, but refused by pharma companies, and they can do it. So what about orphan regulation? It doesn't work here. And what I hear is a sacrilege, blasphemy. Pediatric cancer is not a fixed rare disease. It's evolving biological system. And in metabolic diseases, in orphan world, we treat stable defect. In cancer, we chase an adapting enemy. Orphan regulation was built for something else than tumor evolution. And pediatric oncology needs more than orphan drug incentives. PRAME, glypican, recently discussed inside the EMA, refused by different companies, and being developed by academy. We need public co-funding, academic trial infrastructure, and more wide support for pediatric oncology. Just this is a survival standard, survival for relapse refractory pediatric cancer, and those are our dendritic cell vaccines combined with the metronomic and other kinds of treatment. Much improved survival. So we can conclude that dendritic cells, autologous vaccines as an ATM example, are not new, not the only, but safe, effective, important part of the receipt. And 2 weeks ago, the same target, PRAME, was targeted by academic development, and we were speaking about our dendritic cells in US as an invited lecture. Question is, why do we have to reinvent the wheel as the wheel already exists? And we have one hill, innovative pharma, generic pharma, and we are losing our children in a stormy waters in between them. So there is a to-do list, and I am not optimistic for the new pharma regulation inside the EU for this. We need new business model. And that's all I have. Let me acknowledge the people doing the whole job, patients, their families, our collaborators abroad, and especially our patients.
Yeah, thank you very much, Jaroslav, for your talk and for mentioning all challenges you have if you treat the pediatric cancer patients, because the cancer is really complex disease, maybe slightly differ from, Yeah, maybe slightly differ from the treatment of the monogenic disease. But for both, what is true, and it's my view, because I'm not the clinical oncologist, I'm the clinical pharmacologist, the drug itself is not just the way how to treat the patients, because we are not treating the gene, we are not treating the cancer, we are still treating the patients, and we need to also see many challenges, including the host or whole host organism, including the immunology aspects, pharmacological aspects, and all host-related aspects of the treatment. And especially it's true for the ATMP. So not just the innovation and research will bring the solution, because we really need to have the patients which is treated by such therapies. So what is needed is not only the therapeutic innovation and research innovation, but also innovative way about the thinking. But it was excellently introduced by the Gonzalo Calvo, so I will not repeat it. Research, innovation, biomedicine research, lab research is done now really quickly, fast, but we need to adapt the whole system, including the not only the regulatory mentioned by Gonzalo, but also the access to the therapies, to the such type of the treatment. And it's definitely true for the small patient population with the biological heterogeneity. It's a big challenge. So we are talking today mainly, and we are talking about the rare diseases, ultra-rare diseases, pediatric, of course, rare patients, populations, about the patients who require the personalized therapies. But also, one thing are the number of patients which we need to treat, but the other things is also the access to the treatment. So there are some inequitabilities between the countries. We have the patients which are not be able treated with not excluded, but not allowed to be treated according to inclusion criteria in some conventional clinical trials and so on. So there is not the gap, but there are many challenges. And I see, and it's my personal view, that for the academic developers, academic producers, and academic society itself, it's a challenge, but it's also the opportunity. We are not driven by the financial profit and income or stockholders, because we are the academics. On the other hand, there are many challenges, because we need to stronger not only the collaboration, but also the knowledge in terms of regulatory knowledge, patient access knowledge to the new innovative treatment and so on. But also it was mentioned by Jaroslav that not only the single drug approach is always something what is the best option for the patients, especially in cancer. We need to thinking like the treatment approach on the single drug approach. So what is mentioned here is that the academia or for academia, this century is really challenging. On the other hand, we have the opportunity to bring something from the lab to the patients, but we need to go through the way. Clinical trials are a part of whole journey for ATMP be accessible for the patients. ECRIN has a network of countries and network of infrastructure supporting the non-commercial academic clinical trials. And we in Czech Republic check note of ECRIN, and also establishing the Center of Excellence for ATMPs called CREATIC, mentioning by our minister as well. We discussed it with Jacques, and we decided to create for ECRIN something like ATMP hub for ECRIN clinical trials, and the Czech Republic and ECRIN and CREATIC would serve as a node and methodological hub for the non-commercial ATMP clinical trials, with the long-term vision to really establish and dedicate ECRIN for the framework for the international clinical trials. And our, let's say, dream is and it's also my personalized view, and it was asked by Jürgen Kubal, if we need just a monocentric trial or if we need a multicentric collaboration. My personal view is that at least for some therapies and development, we also could have some multi-centric, multinational collaboration, but we need to create some framework for this, and this is something what is not on table. We need to discuss it, and ECRIN could play some role, important methodological role to support the multinational clinical trials and as well. Why in Czech Republic and why CREATIC and ECRIN? Because as we said, at the Masaryk University, we established the ATMP Center of Excellence, which build the end-to-end ecosystem for academic ATMPs. We are, of course, we have the patient as our final user, let's say. So we are starting to thinking from the patient perspective what is needed, and then we developed the similarly, but let's say 20 years later, the similar ecosystem within Masaryk University. So we have not only the researchers and innovators, but we have also the people and colleagues with the regulatory knowledge. We have the GMP manufacturing facility, and we try to really go through whole pathway for at least cell therapies and CAR therapies. Mentioned by Jaroslav, and we collaborate for more than 20 years. We established not established, but we developed the cell-based therapies called Pediatrics. We realized phase 1, phase 2 clinical trials, and also finally in Czech Republic, for the first time, we received the hospital exemption, which was approved by our regulatory agency, SÚKL. And we are really proud of this, and we collaborate closely with our regulator to really cover all aspects, which was mentioned by Gonzalo, because we also were needed to submit whole, not the whole dossier, but at least the dossier with the preclinical and the phase 1, phase 2 clinical trial data. And as was mentioned again, the hospital exemption is a national pathway to access for the ATMPs. So my final slide. So I see, and we see, and this is why we in ECRIN decided to have this topic for today, ICTD Day. We see the emerging role of academia, especially in the ATMP cell gene therapies. We need to collaborate and to share the knowledge. Of course, each country has slightly different, let's say, maybe ecosystem, including the state budget to support such innovative approaches, but at least for the clinical trials and regulatory pathway, we need to share our knowledge. So I believe in collaboration, and especially for ECRIN and clinical trials part for the cell gene therapies is the time to bring also some new innovative thinking. Thank you very much.
Thank you very much, Dr. Demlová. Please stay at the stage, because now we have approximately 7 minutes to joint discussion for Professor Štěrba and Dr. Demlová. Again, if you have any questions, please feel free to raise your hand, or we can read some question online if we have. Okay, I see first hand raised.
Thank you, and thank you for a very insightful presentation. I have a question for Regina. Since you mentioned then that every protocol is having patient in mind, what is the procedure of maybe patient engagement and including patients in the beginning of the protocols? Thank you.
Yeah, honestly saying, we are, especially in CREATIC and ECRIN, we are starting with these activities. We decided with Lenka to create something like patient public involvement unit, let's say, just to really invite the patients to at least check maybe the informed consent, main inclusion criteria for the clinical trials, maybe also check the whole, blood draw, which if it's needed or not. So there's a many questions which we can ask with the patients who will be not now, but we plan to do and prepare the environment for this year, and then we will continue with such activities. So definitely, I believe that the patient and maybe.
We have already a parents group to discuss, and they are in the middle of discussions with us for the new protocol for dendritic cells.
Yeah. But the concept itself is now created, and we want to invite really more and more patients, which are partly trained, definitely, but sometimes also the naive patients without such regulatory knowledge is also helpful, because he or she is able to run through the proposal of the protocol and so on.
Thank you very much for answers. And do we have another questions from the audience or online? Okay, in this case, we will save some time, and thank you both for your great presentations. Thank you very much.